Dear fellow PhD students, ladies and gentlemen, it's a pleasure to present on the third progress report. Today I'm going to talk about the effects of micro nanoplastics on health. My name is Nicolaus Sándor. Oh, sorry, I have to step for myself, right? Former central banker and health economist, and a first-year PhD student. The vision of the PhD is very simple. I envision a world which is free of environmental factors which are deteriorating health. And I want to provide the necessary high-quality evidence for this to make it a little easier to happen. The first project investigates the effects of micro nanoplastics on health in animal models, and the second is investigating the effects in human tissue samples. Let's talk about the first one briefly. And let me elaborate on the background, why is this important, why we have to talk about microplastics, right? 9.2 billion tons of plastics has been produced since the 1950s, and more than half of it has been produced since 2004. That's quite a lot. But to put it in numbers which are relevant for humans, we eat around 5 grams of plastic a week. This estimation varies a lot based on demographics and socioeconomic status, but it comes to around a quarter of a kilogram a year. That's a lot. And it has bad endocrine disrupting properties. It reduces fertility, for example. In a time where we have a demographic decline, we definitely should look into all the factors and eliminate as much as we can. Speaking about reproductive health, what can we see on this picture? On the one with the little green dots, that's human sperm, and the green is microplastics attached to the human sperm. On the picture next to it, we can see a human ovary, where we see visible microplastic particles too. That means that right at the point where we get conceived, we also partly containing plastic, right? That is incredible. And during our lifetime, it only increases more and it becomes more. So, what do we do? How do we deal with this? The clinical question is, what is the effect of MMPs on the male reproductive health of mice, and how does the effect depend on the properties and the dose of MMPs? Why male mice and why male reproductive health? Mouse model is the most representative for male reproductive health, and it's not as good for females because the cycle is different. But I'm sure everyone except me is a doctor, so I don't have to bother you with these basics. The PICO is population is mice, exposure is micro nanoplastics or MMPs, comparator is no MMPs, and the outcomes which we look at is sperm viability, sperm morphology, tubal diameter, inflammatory markers, etc. We hypothesize that MMPs worsen male reproductive health. Surprise, right? Outcomes depending on dose and type and way of entry and chemical composition. What will be the clinical and research implications of this meta-analysis? Well, we hope to be able to make recommendations on environmental MMP levels, where to focus funds and what should be measured regarding MMPs and pollution, and what is the social burden of micro nanoplastics. So far, no Prospero registration has been found on this, and only 3 meta-analyses were published investigating mammals, so we are off to a good track. I spare you the details with the key articles. Here you can see my search key, which resulted in 1,816 articles. This is although a smaller slice of the bigger project. The bigger project resulted in 13,000 articles, of which we now just focus on mice and male reproductive health. It's the same project, but we sliced it up because of Prospero. Prospero found our first project too broad, so therefore we narrowed it down to male reproductive health and mice as population. So after 1,816 articles were found, we removed duplicates, which resulted in 1,167 articles, and those we title abstract selected with a coherence kappa of 0.76, which left us 134 articles. And then again, we did the full text selection, resulting in 87 articles with a coherence kappa of 0.8. 127. Yeah. Briefly, the detailed outcomes again. Sperm viability, sperm count, sperm motility, morphology, testis histopathology. Sperm count and viability are probably the most common markers and also the most representing ones. And oxidative stress and inflammatory markers as secondary outcomes will also be highly interesting. I wish I could show you plots, yet I cannot, unfortunately. We are currently, as you can see, in the following stage of data extraction, which we are at around 90% done, preparing risk of bias. But even though I don't have plots, I would like to later on come back with a couple of fun facts what the studies so far seem to clearly state. And before that, let's talk about the second project. We want to investigate the prevalence of micro-nanoplastics in different human tissue samples. We can see that microplastics have been found in almost any human tissue. How is this possible? We ingest microplastics through food or drinking. We get it through skin contact, as the skin is a semi-permeable membrane, and the small nanoparticles pierce the skin and get absorbed, especially in very uncomfortable areas where we have, what's the name in English? Miri. Glands, thank you. Exactly. So we can see microplastics in the brain, in the reproductive system, in blood, in heart samples, and that's terrible because locally they have a worse effect than throughout the whole body, and wherever they are, they cause inflammation and problems, and they are forever chemicals. Worse than the microplastic sometimes itself is a phthalate or some plasticizer, which makes the plastic properties different, which are even more endocrine disruptors. So this is a little terrorist coming in our body and causing a lot of problem. What we want to do is, we want to know what is the prevalence and distribution of microplastics in human tissue samples and how does this vary across different demographic factors. We chose the COCOPAC framework of micro-nanoplastics, tissue samples, and humans. Our hypothesis is that MMP's prevalence differ among human tissue samples. And we hope to add to the scientific literature, and the implications should be showing tissues most affected by MMPs, enabling developed targeted treatment for these different organs or organ groups, and mapping the overall effect. And so far we established that the project is feasible, but we focus on the first project now after long, tedious battling with Prospero for registration. Before I finish though, I would like to give you a couple of fun facts on what you can do against something which you can't do anything against, right? We get microplastics through bottled water, we get it through soup cans and legumes, food is full with it. These are the three most common points of entry to the body. Clothing, nylon, underwear, very bad, for example. But what seems to help is royal jelly so far. A couple of studies have been found that royal jelly of the bee, so in Hungarian it's called mépor, seems to reduce the inflammatory effects on the reproductive health in male and in females. And further, sulforaphane, which is a commonly found substance in green vegetables, for example in broccoli, also seems to reduce the overall effect, not just on the reproductive system, but generally speaking. Yeah. So till I find out how bad it is, please eat a lot of honey and broccoli. Thank you. I would like to close my presentation with this quote: "We cannot solve problems with the kind of thinking we employed when we came up with them." Thank you for your kind attention.
Any questions from the floor?
Don't be shy, guys.
Okay. As an economist, what stocks should I be investing in? Joking aside, it's good to see that you are interested in healthcare. My first question is, why don't you choose a more, for example, cost effectiveness? As an economist, this may be a better approach from your side. Is there
that would be a very intrinsic idea, right? To go directly to something cost related, but the literature is very unclear and many confounders can be found. So our idea was we start to map how bad it actually is and try to quantify it, and then we can put some demographic trends on it and try to isolate how much that costs us then in the end. We can always put a cost analysis on anything, but we have to have the hard data which we have to fish out. See, since demographic decline is a big issue, we can directly calculate how much effect it will have on the GDP, for example, or Gini. But if we can cut out the slice and look at it, is it about endocrine disrupting chemicals which cause this, or is it just a social behavior kind of thing? So we thought we first try to find the biggest fish and then we try to catch it.
I don't familiarize with how to detect nano and microplastic. Is there conventional methods? Is it available, or it can be some biases arise from the methodological that the studies use?
Very good question. Generally speaking, usually it is done with fluorescent microplastics in the animal models, for example, where they have this glowing microplastics, they ingest it into the mouse via oral gavage, for example, and then they look at it with Raman spectroscopy or different ultrasound examinations. There are many, many ways to detect these fluorescent microplastics. I could name around 5, 2 which I just did, and the other 3 which is too medical for me. But those seem to be the standard ways.
Is there any calculation on if we would like to change our behavior of manufacturing plastics, and how much money should be investing in to change the plastic age into a better future?
Can you please reframe the question? I'm not sure if I'm getting it right.
How much money do we need to change the plastic manufacture to be in a better health?
Oh, well, you just found the big fish which I'm hunting, my Moby Dick, basically. I wish I could answer with an exact number, but I cannot. It is a ridiculously big industry. I mean, everything around us, even this thing is out of plastic, right? I don't think it will be substitutable. Even biodegradable plastic, for example, has almost the same effects as normal plastic on the human body. So we really have to look at the use cases where we can keep plastic and where we cannot. The only thing so far what we can do is limit our own intake. I don't think it will be ever possible in our lifetimes to absolutely substitute plastic to something, since it's super usable. I mean, it's probably one of the most moldable substances on the planet.
Okay, thank you. Is there any questions?
Yeah, I have just one question.
Okay.
So, you said we ingest, like, how much was it, 1/4 kilogram a year or something? No,
yeah, around that, depending on where you live. Asia is much worse off. Fish is also a big intake factor.
Okay. We live a long time, longer than a year. Where does it all accumulate? Does it leave the body? Or?
Oh, yeah. See, I wanted to do, for the next progress report, I want to do a human, maybe myself, where you can see how much of you is plastic. I thought that's a funny gig. It accumulates all over your body, in almost every tissue. What we see so far, there are two places which it really, really seems to like in the body. First of all is your testes and ovaries, or general reproductive system, and the brain. And that's not too good, because those are probably the most valuable parts of your body.
Thanks for the answer.
My pleasure. But we also pee out some, and through fecal excretion we also lose some. Yeah, it does.
Congratulations. My question is that how can labs grow plastic-free mouses if microplastic is everywhere?
Very, very good question. There is a standard of laboratory mice breeding. Usually it is, the mice in the studies are usually acquired somewhere in China or from Thermo Fisher or something, you know, one of these experimental suppliers. They usually use hyper-sterilized rooms for breeding, and they can, for example, the benefit with mice is that they have very quick generational switches, right? So within 8 weeks we can have another generation of mice. And therefore you can limit it to a point where it's practically not present. If the room is hyper-sterilized, if you don't use any equipment which contains plastic, then you can create microplastic-free mice. Let me give you another fun fact. The transgenerational effect of microplastics is bad, though. So if pregnant mice were ingested with microplastics, the offspring's health was way worse and permanently. So you couldn't, the baseline of testosterone and reproduction and pups' health quality was below the normal baseline of the previous generation and irreversible, really.
Thanks.